Hey guys, welcome to CakeWalk Cambridge. I'm Aisha and today we're going to start with Chapter 6, Plant Nutrition. So this is a very important chapter. You have a lot of marks that come from this chapter and this chapter is relatively longer than the other ones that we've been doing till now, but it's not difficult at all. Okay, so don't worry about it. If you have any questions, you can always email me. I'm always there to help you guys. So let's get started. Okay, so when you look at the topic, the chapter's name is plant nutrition. Now, the first word that comes to your mind when you say plant nutrition is photosynthesis, because it is the process by which plants manufacture carbohydrates from raw materials using energy from light. So basically, this chapter revolves around photosynthesis and its dynamics. So you need to be extremely thorough with the concept. So first, let's look at the definition. Very often you are asked this for 2 marks. So here I've made bold the parts that you would be getting marks for. So it is the process by which plants manufacture carbohydrates, as you can see in the equation, from raw materials, as you can again see in the equation, using energy from light, as you can see in the equation again. Also, you can't miss out using energy from light because photosynthesis, let's break up the word. Photo means light and synthesis means the building up of molecules, of larger complex molecules from smaller simpler substances using energy and enzymes or catalysts. So energy from light is crucial to your answer. Okay, so as we've studied before in plant cells in chapter 2, you have chloroplasts present in palisade layer cells. So these chloroplasts absorb light energy from the sun and they convert it to chemical energy. If you've done physics before, you will know that chemical energy is the energy that is stored in food and fuel. So the chloroplast converts the light energy to chemical energy, and that energy is used to bring about photosynthesis, as you can see from the definition. So now this third point is basically from an MCQ. Sugar or glucose are the first initial products formed in the leaf, and then they are converted to starch, which is stored in chloroplasts. So I've got this MCQ in many papers, in past papers or topic-wise papers, where the question is, what is the initial product of photosynthesis? And they give you starch, water, sugar, sucrose or glucose, and something else. Your answer is sugar or glucose. That's your answer. It's the first product formed. Now let's look at the equations. You're expected to know both of these, and this should be the one thing that comes to your mind immediately. Like if I wake you up in midnight, you have to be able to state the equation, both word and symbol. So the word equation is your core level, which is carbon dioxide plus water in the presence of light and chlorophyll, gives glucose plus oxygen. And in the symbol equation, we have 6CO2 plus 6H2O, light and chlorophyll, gives C6H5O6 plus 6O2. So just remember it's 6616. This is the order: carbon dioxide, water, glucose, oxygen, 6616. You have to state the balanced chemical equation. You can't just write CO2 plus H2O gives C6H5O6 plus O2. You write 6 because it has to be balanced. They will always ask you to write a balanced equation. And if they don't write a balanced equation, you're still expected to write a balanced equation because that will give you a mark. Okay, carbohydrates. You're expected to know what carbohydrates are used for. So the glucose that is formed can form chains to form chains of a polymer, which is cellulose. Because glucose is a small repeating subunit, so it's a monomer. So when it forms chains of glucose, it forms a polymer, which is cellulose. Cellulose, as discussed earlier in chapter 2, is used to make cell walls, which is a boundary around the cell. And it is not selective. So everything passes through it and goes up. Then you have carbohydrates, which are used in the form of storage, that is starch. Starch is stored, and then in spring, where the leaves are like not grown yet, where they're going to begin to form, the starch which is stored is then transported to use energy. It is then converted to glucose and then used for energy. Now, glucose for energy comes from respiration. So your respiration formula is basically the opposite of photosynthesis, but you eradicate light and chlorophyll from the equation. So it's glucose plus oxygen gives you carbon dioxide plus water. You will also do it in chapter 11 and 12, but I think you should know this now. So again, it's 6O2 plus C6H12O6 gives 6CO2 plus 6H2O plus energy. And then another important element that is overlooked sometimes and not made note of is fructose and sucrose in nectar. Fructose is used for nectar, and that is secreted by the plant in the nectaries. So if you mention this, you get your mark. You should know at least 3 of the 4 uses, but I recommend that you do all 4 like thoroughly. You should always be thorough with your concepts. Don't leave anything to chance. Okay, photosynthesis requirements. As you've seen from the definition, you need light, chlorophyll, and carbon dioxide. Now, how do you investigate if you need it or not? Like, how did scientists first know what is required? So, for these experiments, you so for these kind of tests, you carry out experiments in which the component that is being studied is going to be changed. So, for example, if we have light, photosynthesis requirements light, the lights that are going to be used can be controlled by a lamp. So you have one plant that is in the presence of light and one that is in the dark, and then you compare where does photosynthesis occur. If it occurs in the plant only, if it only occurs with the plant in the light, then it shows light is a requirement. So the same way goes for chlorophyll and carbon dioxide. I will explain all of these to you, but these are just basic pointers you need to know before we delve deeper into the concept. Okay, now just before we move on, there's a word in science and bio which is control. Now, control can either be keeping all variables same except one, that is controlling the conditions, which you are asked in ATPs about. And the second type is a control, which we will do later. So it's basically having a setup with the factor that is being investigated not present, to show that no other overlooked external factor is influencing the results. So it's later also as well in this slide, but basically it is to show that only the factor being investigated is going to have an impact on the results and no other overlooked factor. Okay, so just know this term right now, it's in your textbook, but yeah. So light is controlled by lamps. Chlorophyll can be controlled by a variegated leaf, which is basically a leaf with some patches of green and some patches of white. The patches of green have chloroplast, so chlorophyll is present there, and the patches of white have no chloroplast, so no chlorophyll is present there. And then the third component is carbon dioxide, which is controlled by sodium hydrogen carbonate and soda lime. Sodium hydrogen carbonate is a chemical that releases carbon dioxide, and soda lime absorbs carbon dioxide. So these two are also very important in chapter 11 and 12, where you will study respiration. Okay, so let's move on. Okay, so basic test for starch. Now, as I told you in chapter 4, we had the starch iodine test where you add iodine to a sample that's allegedly containing starch, and a blue-black color means starch is present and a yellow-brown means starch is not present. So here the result color is the same, but the procedure is a little different because here you're testing with a plant leaf. So you use forceps and you dip a leaf into hot water for some time. So this softens the leaf and denatures membranes because the hot water again has an effect on proteins as spoken to you about in chapter 5. And the shape of the proteins gets disrupted and the membranes denature, so it makes the cell more permeable. Then after that, you dip the leaf into a test tube that has ethanol. So the ethanol has to be placed in a water bath containing water, like really hot water, and the ethanol has to be boiling a little, so around 78 degrees Celsius. And that causes the chlorophyll to be removed from the leaf. The ethanol opens up pores and drains the chlorophyll from the leaf. Now, why is this necessary? Because when the leaf is white without chlorophyll, any color change is going to be much easier to see. Okay, then you dip the leaf back into water, and then after a few minutes, you take it out, put it on a white tile. Now, why white tile? Again, so the color change can be visible. You place the leaf on a white tile and you add a drop of iodine to it using a dropper. A blue-black color means starch is present and a yellow-brown color means starch is not present. So usually you would do this experiment in school, but because of COVID, I'm not really sure what you're going to do. But I hope this explanation is a little easy. You can always use videos online, YouTube videos. I'll try and find someone, paste the link down below. But I hope this helped you. Okay, so now we're gonna start investigating the factors, the three factors: light, carbon dioxide, and chlorophyll on photosynthesis. So how do you show that light is required for photosynthesis? First, your first step is to destarch a plant. How do you do it? You place it in the dark for like around 3 to 4 days so photosynthesis can't occur and all the starch is used from its reserves as energy, as a source of energy. Okay, so the starch is removed completely. Then after that, from the leaf, on the plant, you cover one leaf with aluminium foil completely to shut it out from the dark, conceal it from the light, and you leave the other one open, uncovered. You place the plant in bright light, and after 3 days, you carry out the same starch iodine experiment in the earlier slide. The leaf in the light, which is uncovered, would give a blue-black color because light is required for photosynthesis, and the covered leaf will give a yellow-brown color because light is necessary for photosynthesis. So no light means photosynthesis can't occur, starch can't be formed because glucose is not formed. Okay, next, chlorophyll and photosynthesis. This is a really important concept and I've even pasted an MCQ question below. So again, you first destarch the plant and then you use a plant with variegated leaves. Now variegated leaves means white areas as seen here and green areas as labeled here. So what you do is you destarch the plant, then you leave the plant in the light for a few days, and then after a few days you carry out the starch iodine test. Now when this happens, the green portion which has chlorophyll and chloroplast is going to be blue-black, and the white area or the yellow area, depending on what the question is going to be yellow-brown. Why? Because the white area has no chlorophyll, has no chloroplast, and so photosynthesis can't take place. Because A, photosynthesis occurs in the chloroplast, and B, photosynthesis requires chlorophyll. So because both these conditions are obviously not met, the white area can't photosynthesize, and so it's going to be yellow-brown. So if this is your question, your answer after the starch iodine test would be C. The green area will turn blue-black and the white area will be yellow-brown, which is shown by leaving it blank. Then after that, we have carbon dioxide and photosynthesis. I explained before, soda lime removes carbon dioxide from the air and sodium hydrogen carbonate adds carbon dioxide to the air. So what you basically do is, you have a jar, okay, or you tie a polythene bag around a plant, okay, and in the polythene bag you have a small dish with soda lime in one scenario, and in the another scenario with the same plant you add sodium hydrogen carbonate instead of soda lime. After a few days you carry out the starch iodine test, and the setup which has sodium hydrogen carbonate is going to be blue-black color, the leaves over there. And with soda lime, it's going to be yellow-brown because soda lime absorbs carbon dioxide from the air. So there's no carbon dioxide that will diffuse into the leaves of the plant. So photosynthesis can't occur because carbon dioxide is a requirement for photosynthesis. So you can either use like a jar like this, okay, but make sure both plants are under light, or you use a transparent polythene bag, transparent being the key word here, because light has to enter. Like you can't leave out another factor if you're investigating one factor. If you're investigating carbon dioxide, every other factor has to be present in the right proportions. If you're investigating light, carbon dioxide has to be present. They're all dependent on each other. Okay, now we're going to go on to the next concept, the rate of photosynthesis. Now, the rate of photosynthesis can be measured by counting the bubbles of oxygen produced or the volume of the bubbles of oxygen or the volume of oxygen produced. So you can count the bubbles of oxygen by manually counting, or you can count the volume by using a gas syringe and delivery tube, which is a lot more work. But yeah, so let's look at the bubble method right now. So rate of photosynthesis is affected by three factors. One is light intensity, intensity being the key word, light intensity, temperature, and carbon dioxide concentration. Carbon dioxide concentration being the key word for this part. light intensity, temperature, and carbon dioxide. So this is an example of how you measure the rate of photosynthesis using different light intensities. So you have a bulb, okay, from a stand, and you place a glass shield, a glass block in between the setup and the light. Now, why? They've asked it this before. So this is because it absorbs some of the heat. So only like the light goes through, not the heat. You know, you don't want to have that excess heat because then you're messing with the temperature as well. So this absorbs the heat and protects the plant because high temperatures can damage the plant because high temperatures damage proteins, and proteins are essential for growth and reactions as proteins make enzymes. I hope I'm not going too fast, but that was that. So you use a glass block. Now to investigate light intensity, you vary the distance of the light source of the lamp from the setup. So a high light intensity would be a lamp which is 10 centimeters away from the setup. A moderate light intensity would be one which is 20 centimeters away from the setup, and a low light intensity one would be where the light is 30 centimeters away from the setup. So the glass block, and then you start a timer as soon as the setup is arranged. This is Canadian pondweed for IGCSE. This is not the same, but usually in IGCSE we use water plants for this kind of experiment because they respire in water and you can count the bubbles. So you use Canadian pondweed, you place a funnel over it and then a test tube, and then you count the the bubbles of gas being produced in a certain number of time using a stopwatch. So the rate is given. So you divide the number of bubbles per time in seconds because seconds is the SI unit for time, and your unit would be bubbles per second or bubbles per minute. So that's for your light. For temperature, you use a thermostat or a water bath and you change the temperature accordingly. And for carbon dioxide concentration, you use chemicals like sodium hydrogen carbonate and soda lime once again to vary the amount of carbon dioxide being produced. There are many chemicals. Usually they give you which chemical can be used, but that was that. And then you count the number of bubbles per time. Now, a limiting factor, very, very important concept. I personally hated this concept when I studied my IGCSE, but okay, let's go. So a limiting factor is basically something present in the environment in such short supply that it restricts light processes. So this includes light intensity, carbon dioxide concentration, and temperature, which were the three factors that affected the rate of photosynthesis. Because so basically nutrition is a life process, right? Nutrition in plants is brought about by photosynthesis. So here these three factors are limiting factors that affect or restrict photosynthesis, okay? So let's investigate this. Okay, so now these are the graphs you will need to know. So first we're looking at light intensity. So as the light intensity increases, the rate of photosynthesis should increase, right? Because then the plant absorbs more light energy, converts it quicker to chemical energy. And so because of the greater amount of energy present, glucose is formed faster and more glucose can be formed. Okay, so initially as the light intensity increases, the rate of photosynthesis increases. So when the graph is not flat, when it does not level off, when it's still sloping, the factor on the x-axis is the limiting factor. So till here, around here, that they've marked till here, where it's not flat, light intensity is the limiting factor. Because a decrease in light intensity will cause a decrease in photosynthesis, and an increase in light intensity will cause an increase in photosynthesis. However, at 38%, where the graph levels off, important term to use in your answers, levels off or flattens, it is not the limiting factor anymore, which means more light intensity is not going to increase the rate of photosynthesis and less light intensity will not decrease the rate of photosynthesis. It has leveled off. Okay, so when the graph levels off, light intensity is not the limiting factor, but temperature, carbon dioxide concentration, or even the amount of chloroplasts available could be limiting factors. So very often they'll ask you to describe a graph and explain why it happens. So you have to mention that before it levels off and it's still sloping, light intensity or whatever the factor is the limiting factor. Once it levels off, light intensity is not the limiting factor. If you state that, you get a mark. And then if you state what could be the limiting factor, you get another mark. So always make sure you know your backups. Okay, now temperature. Temperature is a little different actually. So increasing or decreasing the optimum temperature will result in lower rates of photosynthesis. So the optimum temperature is between 25 and 35 degrees Celsius. Now, why is there an optimum temperature? Because the plant comprises proteins which make up enzymes, and these enzymes only operate best at optimum temperatures. Higher and lower higher will cause them to be denatured, lower will cause them to be inactivated. So that's why you need to so the rate of photosynthesis will increase because it's reaching its optimum temperature, and then it will decrease because it's moving away from the optimum temperature. Then you have your carbon dioxide concentration, which is similar to your light intensity. So when the graph is sloping, carbon dioxide concentration is your limiting factor. And then when it flattens or levels off, it's not your limiting factor. Light intensity and temperature could be your limiting factor because carbon dioxide concentration has no effect on photosynthesis after it levels off. Okay? Okay, so this is like a simplified diagram. So light intensity and rate of photosynthesis. First, it increases proportionally. Okay, when you're looking at proportionality in any science, I just have to bring this up. It means the graph has a straight line that goes through the origin. You get 2 points for mentioning both points. Okay, so it's a straight line going through the origin, so it's directly proportional. And then after that, the rate of photosynthesis becomes constant. Why? Because the limiting factor isn't light intensity anymore. The graph has flattened off, and so an increase in carbon dioxide concentration or temperature will be required to raise the rate of photosynthesis even more. So they become the limiting factors. Okay. I hope I made this clear to you guys. Okay. Now this is using greenhouses and glasshouses. You use them so in greenhouses, you have the use of carbon dioxide enrichment, which is brought about by carbon dioxide cylinders, artificial lighting, that is all your white LEDs and lamps, that increase the rate of photosynthesis because light is a limiting factor. Then you have thermostats or temperature controls that regulate the temperature, and then you have ventilation, which regulates the movement of air in the glass house. Okay, so gases move in and out of the leaf, so it just regulates the air, the wind movements in the glasshouse. You should be able to state all four. It's come before for a 4-mark question. So, okay, the hydrogen carbonate indicator is one that many students mess up with and can't remember color changes, but I'll give you my trick here. Okay, so basically hydrogen carbonate indicator is an indicator that tells you the carbon dioxide content present in an environment. Okay, so it's a liquid. So when it's orange, it means that there is no orange color means that there is no change, orange or red, whatever you want to call it. It means that there is no carbon dioxide. It's a control. There's like absolutely no. It's a normal level. Okay, it's the normal level of carbon dioxide. Normal, like it's normal. It's like what's present in the air that is 0.04%, that would be orange color or red. Now this is a control. Here you see there are no leaves present, no external carbon dioxide present, no added on elements present. So it's a control, a biological control, what I was talking about. Okay, then hydrogen carbonator is usually purple. In our textbook, in our syllabus, it's labeled purple, not red. So it's usually purple when there is a low carbon dioxide concentration present, and it is yellow when there's a high carbon dioxide concentration present. Now, how did I remember which color it was where? So basically, I had this thing in my mind, P for photosynthesis, P for purple. So when a plant is photosynthesizing, the indicator is going to turn purple because when it's photosynthesizing, it is taking in carbon dioxide. Okay, it is absorbing carbon dioxide and photosynthesizing. So carbon dioxide decreases in the cell. So P for photosynthesis, P for purple. And then the other one was respiration and yellow. So when you respire, you release carbon dioxide as according to the equation. And so the indicator becomes yellow. And as you can see here, you know if a plant is photosynthesizing or respire. So a plant always respires. Never get that wrong in an MCQ. Usually they ask you when does a plant respire, and a lot of kids say in the night because in the day only photosynthesis occurs, but that's not true. Respiration occurs from the morning till the night, and photosynthesis only occurs in the morning. So here you know that the plant is photosynthesizing and respiring, but it's going to photosynthesize more because it's in bright light. So the rate of photosynthesis is greater than the rate of respiration. However, here the rate of photosynthesis is less than the rate of respiration because it is dark. There is aluminium foil that has shut out light. It's concealed it from light. So here you know it is only respiring, and this could be considered the compensation point where the rate of photosynthesis is equal to the rate of respiration. That is midday. So if there had to be leaves at midday in this test tube, that would be at the compensation point. Okay, now this is the structure of a leaf. I have cut down this chapter and made it really short because I'm going to talk more than like give you all slides and all, because I just feel it'll help you all this way. A lot of this is mentioned in your textbook. This channel is tailored towards giving you higher marks, not just the understanding. It's like the little tips and tricks to remember a few things. So I thought I'll just go over this really quickly. So the structure of the leaf is very important. You have the waxy cuticle, you have the upper epidermis, you have the palisade mesophyll layer, very important. You have the spongy mesophyll layer, and then you have the lower epidermis. Here you have guard cells. These spaces, these like holes are stomata. You have a waxy cuticle below, and you have air spaces within the spongy mesophyll. So let's go through every part now. First, you should be able to label this. So what I did is I took 3 printouts of this in grade 7 when I first studied it, and I kept labeling every printout, for practice, so I remember. So the waxy cuticles below and above, they protect the plant from water entering the plant. Okay. Then you have the upper epidermis. Its 2 adaptations are thin and transparent to allow light to be absorbed by the leaf. The most light to be absorbed by the leaf. It has no chloroplasts, as you can see. Then you have the palisade layer, palisade mesophyll, which has the most chloroplasts. Whenever they ask you which has the most chloroplasts, is palisade mesophyll layer. So that is where most of the photosynthesis takes place, because more chloroplasts, more photosynthesis. We've done the palisade cell in chapter 2, so you can go back to that chapter and have a look at it just for a revision. Then you have the spongy mesophyll layer, where you have mesophyll cells, which have fewer chloroplasts than the palisade layer cells, because more of the photosynthesis occurs in the palisade layer. You have air spaces in the mesophyll to allow diffusion of gas. So the 2 structural adaptations of spongy mesophyll are air spaces and chloroplasts for photosynthesis and for diffusion of gases. Then after that, you have the lower epidermis, which contains epidermal tissue, and guard cells. Guard cells have chloroplasts as well, and they control the opening and closing of stomata. Okay. Now, when they ask you, I've also mentioned this in chapter 1, I think, when they ask you the order of which cell has the most chloroplasts or which layer has the most chloroplasts, it's palisade, then spongy mesophyll, and then guard cells in that order. Guard cells have the least chloroplasts. Okay, so those were your structures. So now how does photosynthesis take place? Carbon dioxide from the air diffuses into the cell through the stomata from a higher concentration to lower concentration down the concentration gradient. As a result of its random motion, it diffuses through air spaces in the stomata and through cell membranes until it is fixated into the chloroplast. Water is absorbed by the roots via osmosis from the soil from a region of higher water potential to a region of lower water potential. It then travels up the leaf via cohesive forces, which you will learn in chapter 8. When it travels up the leaf, it meets with the carbon dioxide in the chloroplast. In the presence of chlorophyll produced by the chloroplast and energy due to the light, the 2 of them combine and form carbon dioxide and water. I'm sorry, and they form glucose and oxygen. Sorry. Carbon dioxide and water combine and they form glucose and oxygen. The glucose is then rapidly converted to starch, and the oxygen diffuses from the leaf into the air from a higher concentration to lower concentration. You mention all these terms from chapter 3, you get so many marks, okay? So that was basically your process of photosynthesis. Now mineral ions. So mineral ions, so you have nitrate ions, which are used for amino acids. So these amino acids make proteins. Amino acids are the monomers of proteins. So when proteins are formed, enzymes are formed, structures are formed, membranes are formed, DNA is formed. It's like a lot of proteins are usually used for growth. So a lack of nitrate ions causes stunted growth, or the bottom leaves become yellow and the top part remains green, those kind of things. Now magnesium ions are used to produce chlorophyll, so a lack of magnesium ions causes chlorosis. You don't really need to know the term. You can just say it turns yellow from the plant turns yellow from the bottom of the stem upwards. But the term just makes your life much easier. You don't have to explain much. So lack of magnesium ions causes chlorosis. Then you have other elements like sulfates and phosphates. Phosphates are used for DNA. So these elements are called trace elements because they are required in small quantities from the soil itself. Okay, so that was chapter 6. It was a little long, today's video, 30 minutes. I hope you enjoyed it. I hope I got you to understand everything, especially limiting factors. You get 6 more questions based on them. If you have any doubts, please email me. I'll drop down my email below. I'll try and paste a few links about the experiments. And I hope this helped you. Don't worry, we'll get through this together. Bye.